Good morning, everyone. My name is Connie Zhang, a technical support scientist at ACD. Welcome to today's webinar. In the first part of the webinar, I will be discussing the steps for success with RNAscope assay, and then I will be passing the presentation to my colleague Madhura, and she will be talk about the new product development on RNAscope 2.5 HD Duplex Assay in the second part of the webinar. A few points before diving into the webinar. The audience will automatically be muted. If you would like to ask a question, please feel free to select the chat button to type in your questions. If we are unable to address your questions during today's session, we will reach out to you and follow up offline. So I will be covering 6 topics today. However, I will spend quite a few time on troubleshooting tips, the common issues our customers have been experiencing and frequently asked questions. We will then end the webinar with new product development. Let's start with how RNAscope works. RNAscope is a novel in situ hybridization niche technology with unique probe design that allows simultaneous signal amplification and background suppression to achieve single molecule detection in a single cell level while preserving tissue morphology. RNAscope assay works virtually for any genome, any gene, in any tissue. RNAscope assay starts with permeabilized tissue with RNAscope pretreatment kit to allow the target probe to penetrate into the cell to bind to the target sequence. The specific sequence that picked up by the double Z probe pair will then get amplified through a series of amplification cascades, and the signal can then be visualized under the microscope as punctate dots. Each dot represents a single copy of RNA. The signal can then be quantified on a single cell level by manual counting or automated image analysis using different softwares. Then let's talk about the key aspect of RNAscope technology, which is the ZZ probe design. Each Z contains 3 elements. The lower region of the Z is an 18 to 25 base sequence that is complementary to the target RNA sequence. The upper region is a 14 base tail sequence, a spacer sequence in between that links the both upper and the lower region of the Z. The double ZZ probe makes up the target-specific binding site that is 15 base region, and then the upper region of the double Z is the pre-amplifier binding site, and is where the amplification cascade occurs. With a standard probe design, the target probe set is a pool of 20 ZZ oligo pairs, and it binds to a 1 kb target RNA target region. Oftentimes, our customers are working with samples with degraded RNA. Our probe can also bind to fragmented RNA to generate an RNA sequence to be visualized as punctate dots. With the unique ZZ probe design, mRNA transcript detection of RNAscope assay is based on highly specific and robust signal amplification. There are 4 different types of manual assay to choose from, including chromogenic brown, red, and duplex, as well as the multiplex fluorescent assay. The first 3 assays are chromogenic assay and based on either the HRP and AP chemistry. The multiplex fluorescent assay is designed to detect up to 3 targets in one assay, and the fluorophore that comes in wavelengths close to 60, size 3, and size 5. For a single-plex chromogenic assay, only ready-to-use C1 channel probe are used, whereas for the duplex and multiplex fluorescent assay, C2 and C3 probe will be used in combination with C1 probe. This slide highlights the amplification steps of the brown and red chromogenic assay. The amplification cascade starts with pre-amplifiers binds to the tail sequence of the ZZ probe pair, and then followed by amplifier and label probe binding. The label probe conjugate with either the HRP or the AP enzyme. By reacting with DAB substrate or phosphorid substrate, a brown or red signal will be developed. So a few points to note here. We do not recommend interchanging reagents within brown and red assay or cross similar 2.5 HD assay. By default, 2.5 HD assay requires C1 probe that are ready to use. No further dilution is required. So for a duplex assay, the amplification cascade occurs simultaneously for the C1 probe as well as the C2 channel probe. Here for the C1 channel probe, by reacting with the green substrate, a green signal will be developed, whereas for the C2 probe, by reacting with phosphorid, using the AP chemistry, the red signal will be developed. So please pay attention to the following tips here. By default, C1 probes are ready to use, comes in 1x concentration, whereas the C2 probes are in 50x dilution, 50x stock. So to make a duplex probe mixture, at 1x concentration, you will need to mix the C2 probe 1:50 dilution with the C1 probe. It's also possible to use the C2 probe alone. You will need to use the blank probe C1 as a probe diluent and mix the C2 probe in 1:50 dilution in the probe diluent. Moving on to multiplex fluorescent assay. The label probe is now tagged with 3 different fluorophores, which can be detected in different fluorescent channels, which are the Alexa 488, Auto 550, and Auto 647. So here is the recommended excitation and emission wavelength to visualize your multiplex fluorescent signals. And similar to the duplex assay, we want to again point out the tips here. The C1 probe is ready to use, whereas the C2 and C3 probes are in 50x concentrated form. And to make a 3-plex probe cocktail, you will need to mix the C2 and C3 probe diluted in 1:50 dilution with C1 probe, again using the blank probe C1 if you only want to use the C2 and C3 probe only. So the chromogenic assay consists of 4 key parts: pretreatment, hybridization, signal amplification, and detection. So the whole workflow takes about 7 hours. Using FFPE sample as an example here, the pretreatment contains deparaffinization H2O2 step to block the endogenous peroxidase activity, followed by epitope retrieval to break up the cross-linking that is introduced by the fixation step, and then the protease digestion step to penetrate the cell, to permeabilize the cell, to make sure that the probe can penetrate to bind to the target sequence. After the pretreatment, it's followed by target probe, preamplifier, amplifier hybridization, and finally label probe hybridization. Then you will be doing a hemotoxin counter stain and finally imaging the signals using light microscope. So if you are using sample types other than FFPE samples when running chromogenic assay, please go to this website for the appropriate user manuals for your specific sample type. For multiplex fluorescent assay, the workflow is very similar to chromogenic assay. It contains the same 4 main parts shown here. Using fresh frozen sample as an example, the pretreatment start with a brief fixation and followed by dehydration, protease digestion, and then you will go through the 4 main steps here, target probe, preamplifier, amplifier, and label probe hybridization. The only difference here is you will be doing a DAPI counterstain, and then the image will be visualized by fluorescent microscope. So again, working with samples other than fresh frozen with multiplex fluorescent assay, please go to this website to find the right user manual. So to provide flexibility to our customers, RNAscope assay is designed in a way that can be performed in either 1 day or split into 2 days. So if you would like to split the assay into 2 days, the stopping point would be after the sample pretreatment step. So please review the user manual part 1 and part 2 for detailed optional stopping points. Okay, let's now move on to tools for getting started. Here's a list of, here's the checklist we highly recommend customers to go through before starting our RNAscope assay. And please make sure that you have all the must-haves listed in the checklist to ensure a successful RNAscope assay. And for more information to get started, please go to this website, and then checklist is also available on the website and in the user manual. Here is a typical setup for a hot plate to do the target retrieval step. It seems very simple, but it's a very important step in RNAscope assay. So when using the hot plate, we highly recommend monitoring the boiling temperature and time during the entire boiling step. Please maintain the temperature at around 100 degrees C and monitor the temperature using a thermometer. You can also use a steamer for this target retrieval step. Please refer to the user manual for more detailed information. The RNAscope reagent kit consists of 3 subkits, including the pretreatment reagents, RNAscope detection kit, and wash buffer. And it's very important to note that our probe needs to be pre-warmed at 40 degrees C for 10 minutes before use. And if any precipitation is present, please warm the 50X wash buffer at 40 degrees C for 20 minutes before diluting it. Moving on to HiveEasy hybridization system. It comes with the HiveEasy oven, the humidity tray and humidifying paper, and also the flat rack. This oven is designed to maintain the perfect temperature and humidity to ensure optimal and consistent RNAscope staining. It's a very important must-have for successful RNAscope assay. Here are the 2 options you have for washing slides. You can either use the Tissue Tech washing tray, which is very commonly used in the lab, or you can use our easy batch slide processing system, which helps streamline your RNAscope assay and save slide handling time, especially when working with a large number of samples. So moving on to tips and tricks for sample preparation. Here's the recommended sample preparation for FFPE samples. We recommend fixing our sample in 10% NBF for 16 to 32 hours at room temperature as the optimum fixation condition. Over the years, we also learned that not only the sample fixation, but also the sample storage are very important for RNA quality. For FFPE block, we recommend storing at room temperature with desiccant to preserve the RNA quality. If you are working with archive samples, we found the 4-degree storage can preserve the RNA quality even better than room temperature. For FFPE tissue sections, we recommend store at room temperature with desiccants. Please use up your FFPE sections within 3 months for the best RNA quality. One tip to note here is please review the user manual part 1 for sample preparation guideline. So here's an example of successful RNAscope staining using chromogenic red assay on properly fixed and prepared human breast cancer tissue. With proper fixed sample, you can get very strong staining with our positive control PPIB probe and very specific staining with the target probe. And so if you are having any issues getting successful RNAscope staining, please feel free to contact us at support, or you can go to this website for more troubleshooting information. For sample preparation, sample thickness is also very important for detecting signals. So this slide here shows that we have stained different thickness of fresh frozen mouse brain samples with our positive control Polar 2A and PPIB control probes. So as you can see here, the sample thickness varies, and then the signal levels varies with the sample thickness, and the thicker the sample, the more signal you get. So with this data here, it suggests that the sample thickness can also affect signals in your sample. So our recommended sample thickness for fresh frozen is 10 to 20 micron, as for fixed frozen is 7 to 15 micron. Moving on to troubleshooting tips. From a very broad, high level, the general tips for RNAscope manual assay is always follow protocols exactly as described in the user manual. And also, please review the checklist. Make sure that you are using all the required material. And also, always use control probes and slides to qualify your samples. I will go into details on this in the later slides. And also, review the sample pretreatment recommendations. And I will also expand on this in the next few slides. So if you have any questions, please visit this website here for troubleshooting tips and frequently asked questions. The first troubleshooting topics I want to cover is sample qualification. So here's the RNAscope scoring guideline we use to qualify the samples. So we have score from 0 to 4, and the scoring guideline is based on the number of dots per cell and also the presence of clusters. So the first thing you will need to do is a quick technique check using the control HeLa or 3T3 control palette, and stain the control slides with negative and positive control probes, and then this is the expected results you want to see. So if you get a very strong PPIB staining and very clean DAB staining, that means you have passed the technique check, and then you are good to move on to the second step, which is the sample RNA quality check. So here again, you will use the control probe to stain your target tissue, and then this is the expected expression pattern you want to get. So you want to see a strong PPIB staining and then clean DAB. Here is the QC standard you always want to refer back to. And so if your sample passed the QC, please you can go ahead and run your target probe. If your sample failed QC, there are 3 actions to take. First, verify technique. Second, check RNA quality with new samples. Third, perform perform assay optimization. So we always recommend our customers to qualify your samples before staining your samples with any target probe. So after seeing how the successful chromogenic staining look like, now this slide is just to show you how the successful multiplex fluorescent results look like. And so here, I want to emphasize this point before diving into the results. Always use 3-plex positive and negative control probes to qualify your sample and your assay. So here in this experiment, we have used the 3-plex positive control probe, and AMP4A was used. So in the green channel, you can see a low to medium polar-2A expression and medium to high PPIB expression in the auto 550 channel. And you can also see a very high UBC expression in the auto 647 channel. So you can always use a different AMP4 reagents for alternative color combinations. So another troubleshooting topic I want to cover here is sample pretreatment. So first thing you may ask, why do we need to optimize sample pretreatment? So for FFPE samples, the optimal fixation is 10% MBF, 16 to 32 hours at room temperature. Oftentimes our customers are using under-fixed or over-fixed samples, or maybe special sample types listed here, which are sensitive to pretreatment. And you may also use special tissues listed here, which require some pretreatment optimization. So if you are working with any of the samples listed here, please feel free to contact support, and we are more than happy to provide more optimization recommendations. So here is a workflow for sample pretreatment optimization. We always recommend customers starting with the standard pretreatment conditions, and you can find all the standard pretreatment conditions on this website here. And so based on the staining pattern and also the tissue morphology with standard pretreatment condition, we will then decide whether or not the pretreatment optimization is required. If it's required, we will then adjust the pretreatment conditions by adjusting the boiling time of the target retrieval or the protease time. For example, for FFPE samples, the standard pretreatment is 15 minutes target retrieval and 30 minutes protease class. For example, if you are observing high background, that means your sample may have been over-digested. So the solution is to reduce pretreatment condition by dialing the protease down from 30 minutes to 15 minutes. So for example, if you have observed no signal, and that means your sample has been under-digested, so the action to take is increase the pretreatment by increasing the boiling temperature from 15 minutes to 30 minutes. So once again, please refer to this website for recommended standard pretreatment conditions. So this slide summarizes the standard pretreatment conditions for each sample type and also each assay type. So for example, for fresh frozen chromogenic, you will be doing a H2O2 and protease 4 pretreatment. And for multiplex fluorescent, you will be only doing a protease 4 pretreatment. Again, please go to this website for appropriate pretreatment recommendations. Another topic I want to cover here is autofluorescence. So here are some examples of autofluorescence in various FFPE samples. So the green dots you see here is not real RNAscope signal, and it's all attributed to tissue autofluorescence. So one tip to keep in mind, FFPE sample always have inherent autofluorescence. So it's very likely you're going to run into this autofluorescent issues if you work with FFPE samples using a multiplex fluorescent assay. So the solution is use appropriate background correction software to reduce autofluorescence. Now moving on to microscope and sample related issue. If you are observing no signal, there could be either microscope related or sample related. So if it's microscope related, please make sure that you use the correct filter setting. Do not use auto exposure first, and always use a known image enhancing software. So if it's sample related, you want to make sure that your RNA quality is good in the sample, and also you will need to perform a pretreatment optimization. A very common troubleshooting topic is tissue detachment. So if you are working with FFPE samples, we recommend adding additional baking step after deparaffinization and target retrieval if you are experiencing tissue detachment. So general tips for preventing tissue detachment: first, always use Superfrost Plus slides. And second, bake slides in active air circulating oven, not over high-busy oven. Third, maintain mild boiling of target retrieval. Please avoid any bubbling injecting directly onto the tissue, which is one of the main reasons of tissue falling off. So if you are experiencing tissue falling off, please contact support. We are happy to assist you through your specific issue. So if you have tissue falling off issue with fixed frozen, here are the 3 recommendations we give customer. And so the first one, you can extend the air drying step up to 2 hours, or add a post-fixation step after air drying. So the post-fixation step can be 15 minutes to 2 hours. And then also add additional baking step after target retrieval. So if all the 3 recommendations have failed, you can also try skip target retrieval step and extend the protease time from 30 minutes to 45 minutes. Besides all the general tips I have mentioned in the previous slides, please make sure that the sample thickness for fixed frozen is 7 to 15 micron. If you are using thicker sections, it may be another risk factor of tissue detachment. So moving on to frequently asked questions. First, here we want to talk about the important things to keep in mind for red chromogenic assay. So we highly recommend using EcoMount mounting media, since EcoMount is the best to preserve our red RNAscope signal, and it prevents signal from fading over time. And second, do not perform dehydration step with alcohol, because the red signal is very sensitive to alcohol, and any trace of alcohol will cause the signal to diffuse. And finally, samples should be dried in 60-degree oven for at least 15 minutes before mounting. For multiplex fluorescent assay, oftentimes our customers find difficult to understand the different AMP4 options. So here, our reagent kit comes with 3 different options. So when you a rand assay, you will only need to pick one option here based on the colors you want to develop your target probe in. So for example, if you want to see your C1 channel in green color and you want to see your C2 probe in the auto 550 and the C3 in the far red auto 647 channel, you will need to use the AMP4 Alt A. So if you want to try other color combinations, you can use either the AMP4 Alt B or Alt C. So here, another important tip to keep in mind, please use Plum Gold anti-fade mounting media to best preserve your multiplex fluorescence signal. Some additional questions which are often asked by our customers. First, how do I find the user manual? Please go to our website and you will be presented with 3 different filters and select the correct filter, and then you will be presented with appropriate user manuals to use for your specific sample type and assay type. And another question is, what's the key difference between RNAscope assay and IHC for FFPE samples? Although cooling in antigen retrieval is a common practice for IHC assay, no cooling is required for RNAscope assay during epitope retrieval. Users should directly put the slides in water at room temperature, dehydrate, and proceed to the following pretreatment step. One more question, can I perform IHC and ISH dual staining? The answer is yes. We recommend starting with RNAscope first, followed by IHC or IF. So please contact ACD technical support for customer-validated protocol, and please also check out this website for additional information. Here's all the parts for steps for success for RNAscope. Now I'm passing to my colleague Madhura.
Hello, everyone. Thank you, Connie, for that very informative presentation. Now that we have some background on the RNAscope technology, we can now move forward with introducing the new RNAscope 2.5 HD Duplex Assay. It is our pleasure to introduce Advanced Cell Diagnostics' latest product release, the Duplex Reagent Kit. This robust kit was developed based on customer demand and replaces the existing RNAscope Duplex Reagent Kit. The Duplex Kit is a green and red combination using fast red and green. A few key applications of this kit are for the study of gene co-regulation, profile gene expression in a specific cell type, which can be identified using a cell-specific marker, and map co-expression of 2 targets within the same cellular context, based on reliable chromogenic detection of 2 transcripts. Let's review a few important highlights of the RNAscope 2.5 HD Duplex Assay. A few key features are that it's based on dual 2.5 chemistry, which implies that both channels are amplified to the same level. It utilizes a red and green color combination. All the reagents are ready to use, and it requires the same pretreatment reagents as the singleplex assay for FFPE samples. These key features provide the benefits of visualizing a low-expressing target in both channels. It has comparable sensitivity to the 2.5 singleplex assays. It's easy to use, and no additional pretreatment optimization will be required if your samples have already been optimized with the 2.5 singleplex assay. This kit also allows you to have sharper, crisper signal in both channels, and it's easier to quantify and provides better publication quality data. Let's review the reagent kit components. With the manual reagent kit, you will receive target retrieval solution, one of the protease and the hydrogen peroxide reagents, one of the amplification reagent boxes, and one of the wash buffer. As you can note, the kit has currently only been validated for FFPE samples. The fresh frozen sample validation is in progress. Now that we know what the reagent kit contains, let's review the probe product configuration. For the probes, the compatible channels with the duplex reagent kit are the channel 1 and the channel 2, with channel 1 being HRP based and can be visualized in the green channel, and channel 2 being AP based can be visualized in the red. The two channels are relatively comparable in sensitivity, but due to the property of the red color, we recommend to put the lower expressor in the red or the situ channel as it appears to be brighter. So please note, keeping in mind our recommendation for putting the lower expressor in the red channel, we now have appropriate positive control probes with PPIB, which is a moderate to high expressor housekeeping gene target in the C1 channel, and the POLR2A, which is a moderate low expressing housekeeping probe in the C2 channel. Please note, this configuration is different from the existing duplex positive control probe configuration. For the negative control probe, we do have the DAP-B probe in both the channels. For new users, we now have an intro pack with all the must-have materials, as Connie discussed, bundled into a package. An important point to keep in mind here is the use of vector mount mounting medium, and this helps preserve the red and green signal intensity. So as Connie already mentioned, and quickly I would repeat that in the intro pack for the duplex reagent kit, you will get the control slides, you will get the positive control probe, the negative control probe, the reagent kit, the hydrophobic barrier pen, and the vector mount mounting medium. Getting started with the 2.5 duplex assay is fairly easy. We recommend that all new users start with the intro pack, as I mentioned in the previous slide. Also, please ensure that you review the checklist for the 2.5 duplex assay, which is available on the support page, and confirm that all required reagents are present before starting the assay. Also, please review the user manual and the quick guide to familiarize with the workflow prior to starting the experiment. And again, as Connie pointed out, all these documents are available on the support page of the acdbio.com website. So now that we know all about the reagents, the required reagents, and how to get the manuals to run the assay, let's quickly review the workflow for the FFPE samples that you would run if you were to use the 2.5 HD duplex assay. As you can see from the slide, you can start with properly prepared, properly permeabilize the samples, which are fixed with our recommended conditions. The recommended conditions, to reiterate, are 24 hours at room temperature with 10% NBF. This is the recommended fixation condition. This will ensure that the RNA quality is well preserved. So the typical workflow for such samples, which are fixed and embedded in paraffin, is to start with baking for 1 hour at 60 degrees. This step is followed by D-pair and hydrogen peroxide to block any endogenous peroxidase activity. The next step is target retrieval, which breaks the cross-linking that's caused by the formalin fixation and is subsequently followed by the protease digestion. As the RNA is now accessible, probes in channel 1 and 2 in the ratio 1:50 are now hybridized for 2 hours at 40 degrees. Following this, the first channel is amplified to yield the red signal, and then the second channel is amplified to yield the green signal. Now there are multiple stopping points to allow you to run the assay as per your convenience. You may stop after baking, D-pair, and target retrieval for up to 24 hours. It is, however, advisable to complete these steps and stop after the probe hybridization step to split the assay evenly over 2 days. In order to do that, you will require 5x SSC. And lastly, please note that you can split the assay after the probe hybridization step and continue with the channel amplification steps the next day. You will also need to pay special attention to the drying and the mounting steps, and the use of vector mount is critical in order to ensure there is no loss in signal intensity. Now, in order to analyze and quantify your signal, software analysis can be performed with the HALO software, which can provide a cell-by-cell quantitative result. Released by Indica Labs, HALO algorithms are designed for the quantification of complex RNAscope expression data. HALO software provides interactive results with a real-time readout, and the software reports individual cell data and maintains an interactive link between cell metrics and cell imagery. So now quantitative scoring of the HALO image analysis software can be performed for whole tissue evaluation of RNAscope staining. Each cell is assigned to a bin based on the number of dots per cell. Clusters are divided by the typical probe signal area to calculate a dot number for the cluster. An 8-score calculation is performed in HALO to assign a single score to an entire tissue section based on the average target expression in this tissue. So to summarize, the key workflow highlight for the 2.5 HD duplex assay is the ability to split the assay over 2 days. There's an easy transition from the singleplex assay. The pretreatment conditions remain the same. And we do recommend that the lower expressor is placed in the situ channel as the red color is visually stronger. As we are now aware of the product configuration and workflow, let's review a few of the many applications you could use the RNAscope 2.5 HD duplex assay for. This kit has been extensively tested in-house on 76 samples and across several different tissue types, to name a few, such as human TMA, mouse syngeneic tumors, mouse tissues, liver, lung, kidney, xenograft tissues. And we will see examples of the images in the following slides. Now here we list a few of the many potential target applications you could utilize this duplex assay for. We start with the neuroscience application, where you can use this assay for co-localizing a cell type marker to a biological marker of interest. In this example here, NPY neurons are identified and the expression of POMC is studied. This tissue used here is the mouse brain FFPE tissue. In the example of immunotherapy, we see detecting multiple targets of interest, and in particular here we are noticing we are studying FOXP3 and CD4 to characterize a tumor microenvironment. The sample here is FFPE lung cancer tissue. Another potential application is to evaluate different gene expression profiles in patient-derived xenografts. Combined with the specificity of RNAscope probe design, in this example of patient-derived xenograft, we can clearly identify mouse stromal cells and human tumor cells by using species-specific PCAM probes. And here in oncology section, we see an example of studying a ligand and receptor expression in tumor microenvironment. In specific, the detection of NRG1 ligand and its receptor ERBB3 in breast cancer tissue. Let's look into greater detail at an application to understand immunotherapy. Here we present the use of RNAscope for the detection of RNA targets in tumor microenvironment that are involved in tumor immunology and immunotherapy. So specifically, RNAscope duplex assay can be used to identify checkpoint markers such as PD-L1 and immune cell markers such as CD8 alpha. In lung cancer tissue, we see a high abundance of CD8 alpha T cell marker in the tumor and the stroma co-localizing with the PD-L1. However, note in the ovarian cancer tissue, CD8 is restricted only in tumor and stroma, and PD-L1 is only restricted in the immune cells and not tumor. With these applications, let's review some of the data presentation that R&D has put forth for us to review. The R&D did perform an extensive validation of the 2.5 duplex assay. They tested the assay on multiple formalin-fixed paraffin-embedded samples. The samples included cultured cell pellets, tissues from multiple mouse organs, human tumor samples, and human xenografts in mouse. Please note, however, the fresh frozen sample type validation is in progress. We have not yet validated this on cultured cells and PBMCs. In this slide here, we are testing low expressing targets such as TBP and Polar2A on HeLa cell pellets. We observe strong punctate signal in both channels, the red and the green, and no background is observed in the slides when hybridized with the negative control probe. Using the 2.5 HD duplex assay, we stained NRG1 and its receptor ERBB3 in human tumor samples. From the results, we can see in the image to the left, NRG1 and ERBB3 was detected in two different cell populations, the green and the red. In the image to the right, NRG1 was expressed by tumor cells, not the stromal cells. The ERBB3 was detected in the marginal region of the tumor that is adjacent to stroma and in cells in the stroma. Here is another example where we see very specific staining of HGF and its receptor c-MET. We can detect HGF and c-MET in different populations of cells. Another example of co-staining, we used the 2.5 duplex assay to detect the ligand CD248 and its receptor beta-type PDGF receptor in a tumor microenvironment. In the image on the left, you can see co-expression of these two targets within the same cells, as well as in different groups of cells as on the right. And you can see that both these markers are very high expressing. Since they're expressed in different cell populations, there's no masking of one over the other. We also verified the 2.5 HD duplex assay in mouse FFPE tissues. Here we see punctate staining of the two markers, LGR5 and GLI1 in mouse brain and colon tissues. As you can see from this, the 2.5 HD duplex assay can be used to define tissue-specific expression on a single cell level. In the study of xenografts, it has been challenging to separate targets from different species due to high homology between each other. In this case, the tumor cells are human origin growing in the microenvironment of mouse stroma tissue. We designed species-specific probes of targets such as EGFR and PCAM1. Human-specific probe, which is green, only detects EGFR in human-derived tumor. Mouse-specific probe, which is in the red, detect low expression of EGFR in mouse-derived stroma. Another target, endothelial marker, PCAM1, is majorly expressed by stroma cells. Here to the right, we can see red signals of mouse-specific PCAM1 in mouse-derived stroma. Human-specific PCAM1 probe does not generate signal in the mouse tissue. However, please note that the human or mouse-specific probe in xenograph study need to be specially designed such that they have no cross-reactivity with the other species. We also detected similar success in other targets such as FGFR1 and TGF-beta. Using the duplex assay in patient-derived xenograph samples. Again, here we see specific staining of the human probe in the tumor region and the mouse probe in the stromal region. Our R&D team also extensively validated the assay for run-to-run and operator-to-operator consistency in order to provide a robust assay for you, our customers. Let's quickly review the technical details. Here we see an example where we see high consistency between slide to slide within the same run using the same high-VZ oven. As we can see from the images, both the channels, the dots are clear and punctate. Here we see similar results with the assay performance in another high-VZ oven. So it's safe to say that we don't see any variability with the assay between different oven usage. In running the assay over multiple days, we have demonstrated that there is low day-to-day variation. The dot sizes and the signal intensity are consistent even when the assay is performed over multiple days. And here we have demonstrated the consistency of staining with this assay by multiple operators. As you can see from the images here, Again, the dot size and the intensity are both consistent between operator 3 and operator 4. All these consistency studies have been performed using low-expressing targets such as TBP and polar 2A. With these images and the data that I have presented, I would like to conclude that ACD continues with product innovation and now introduces the RNAscope 2.5 HD Duplex Assay. We have demonstrated robust results in different tissue types amongst the different targets, and we have thoroughly tested in-house demonstrating the reproducibility within and between runs, different ovens and operators. In this brief presentation, we have also aimed to provide you with certain applications that you could use our 2.5 HD Duplex Assay for. With this, I would like to hand the presentation back to Connie, who will now conclude this presentation of ours.
Hi, everyone. So finally, we just want to draw your attention to our support page. Please check out this page here for more information. And also, lastly, we are always very happy to assist you with any questions or concerns you may have with regards to your RNAscope assay. Please reach out to us through any of the methods listed here. We look forward to working with you. Thank you very much for attending today's webinar. We will follow up with you on the questions offline. Thank you.